This is Lecture 4, Part E, the last lecture making, or the last file making up Lecture 4. We're going to finish discussing the environment and microbial growth. Let's talk about osmotic stress. All biochemical reactions are water-based, and cells are typically 70% or more water. Thus, for all organisms, including microbes, liquid water is a requirement for life. Water that is associated with solutes is less available for organisms. Therefore, the water that is in any kind of solution is less available to a microbe than the water found in a solution that is just pure water. Water activity is a way of measuring the availability of water to organisms. It is expressed as A subscript W, and it's defined as a ratio of the vapor pressure of a solution. Okay, that solution would be a solution of whatever it is you are interested in measuring, okay, relative to the vapor pressure of pure water. And that's at equilibrium, okay, with those vapor pressures. So therefore the water activity of pure water is going to be 1, and the water activity for anything else will be something between 0 and 1. Shown in this table is the water activity of a variety of substances of interest to humans. Remember that the lower the water activity, the less available the water is to microbial life. Consider that you can see why it's easier for microbes to grow in blood or seawater than it is for them to grow in salami, which is a relatively dry material. Here's salami. It has a water activity of 0.85. Bread, in contrast, has a water activity of 0.95. So bread actually has a lot more available water for microbial growth than salami does. Note the water activity of maple syrup, which is also less than bread. So here's maple syrup. It has a water activity of 0.9, and that's actually quite low, which explains why you very rarely see anything growing in your maple syrup, even if you keep it at room temperature. You don't actually have to keep it in the fridge. This is similar to honey. Honey also has very, very low water activity. It's somewhere in the range of 0.8 to 0.9. So it's very difficult for microbes to grow in honey. Endospores might persist in honey, but they don't go into vegetative cells. They don't grow. Nothing really grows in honey. It's an essentially sterile solution. So the concept of water activity is really important when it comes to the topic of preventing microbial growth in food or other materials. And it's part of the reason we dry some of our foods and salt others or add sugar. This is done to not just for flavoring, but also to preserve our foods. So when microbes are living in very salty environments, very hypertonic environments, they have to have strategies for dealing with that. One of the strategies that many of them use is to synthesize or bring in from the environment materials that are known as compatible solutes. Compatible solutes can be certain amino acids, such as glutamate or proline. They can be certain sugars, such as sucrose or mannitol. They can be amino acid derivatives, such as glycine betaine, or they can be glycerol. And there are other options as well. So microbes will either make these intracellularly or pump them in from the external environment. It's very important that those solutes do not affect cellular metabolism and that they're not toxic in any way. Basically, the idea of a compatible solute is that it is something that a microbe is using to raise the solute concentration intracellularly so that it matches the solute concentration found extracellularly and thereby reduces osmotic stress. This is a strategy commonly used by microbes that prefer high salt or high solute conditions. These are referred to as halophiles. We'll look at those definitions in a minute. A quality of all halophiles is that they can make compatible solutes. So let's describe microbes in terms of their salt tolerances. A halotolerant microbe, and an example here is Staphylococcus aureus. Okay, it can cause blood infection. and other problems. It's a common skin-borne microbe. If it accesses your body when you get a cut and starts to grow, it can cause blood infection, also known as blood poisoning. It's a halotolerant organism. So here's growth rate going up the y-axis, and here's where it grows best, under conditions that are actually fairly moderate or low salt. But notice how far out this curve grows. It can tolerate concentrations of salt equivalent to about 12% sodium chloride and still manage to grow. And this is one of the reasons why it grows well in blood, which is a relatively salty environment, at least compared to a water-based solution. Other organisms, notice this curve here, this blue curve here, that's showing something that would not be very halotolerant at all. And so what's happening there is it grows optimally at about 0% sodium chloride equivalent, and then the growth rate plummets as soon as you increase to 1 to 2% sodium chloride. Halophiles, for example, Alivibrio fisheri, this is a symbiont of the bobtail squid. So it's a marine organism. And if you're a marine organism, you're good with the salinity of salt water, of seawater. So you're typically quite halophilic. You like salty conditions. And in fact, it grows optimally at between about 5 to 7% sodium chloride equivalent. Extreme halophiles, like Halobacterium salinarum, they are able to tolerate salt concentrations, not just tolerate, but actually grow optimally at salt concentrations as high as 20% sodium chloride. That's the point at which you're starting to actually be right at the solubility limit for sodium chloride. So these organisms are found not in routine seawater, but in places like salt flats, highly brine environments. Let's talk now a little bit about the environmental parameter of oxygen, or the lack thereof. So shown here is an equation you should be very familiar with, and if you're not, it's time to start reviewing, because that's something you need to have a solid of an understanding of for microbiology class. That is aerobic respiration. Okay, this is the process whereby glucose, okay, shown here, is oxidized to form CO2. During that process, oxygen is serving as what's known as the terminal electron acceptor. And the oxygen is being reduced to water, okay? And we do that in our cells, and many microbes do that in order to generate ATP, cellular chemical energy. So it's a chemical energy conversion. Now, in the microbial world, there are many more possibilities with regards to what one can use as a terminal electron acceptor. So for example, this equation here, the second equation, shows the oxidation of glucose to CO2 using nitrate. As the terminal electron acceptor. There's no oxygen used in this process. So nitrate is reduced to water, okay, and we're generating ATP in this process, and that would be a type of anaerobic respiration. Okay. Down here we have an equation that if you look carefully you actually should recognize. We discussed this in earlier freshman level biology classes. There's glucose, and it is being oxidized, and it is forming ethanol. Okay, and not written in this equation, but this equation is fermentation, or a type of fermentation, which means that there is an internal electron acceptor, internal terminal electron acceptor, and in this case it's pyruvate. And this is done also to yield ATP. So organisms can make their living or grow doing that, and there's no oxygen present there. So there's no oxygen present in the second or the third equation. So plenty of microbes on Earth are able to grow in the absence of oxygen, or in some cases they're able to do two things. For example, E. coli can use aerobic respiration, and it can also use anaerobic respirations that require nitrate as a terminal electron acceptor, and it in effect can be a switch hitter. Let's think a little bit more about oxygen. So oxygen occurs in many toxic forms. Okay? So in the atmosphere, you can have what is known as singlet oxygen. It's highly reactive and can damage cells through that reactivity. And then in cells, as byproducts of oxygen-based or utilizing metabolism, such as aerobic respiration, cells will generate what are toxic forms of oxygen known as superoxide anion, hydrogen peroxide, and hydroxyl radical. That little symbol right there is indicating radical. So all of those are generated in cells whenever they use molecular oxygen O2 in their cellular metabolism, and they're all really toxic. So in other words, there's a price to pay for using oxygen. All of that ATP that you generate when you use oxygen to do aerobic respiration, you get a lot of ATP out of it, but you pay the price of accumulating a lot of highly toxic forms of oxygen. This would be lethal to cells unless they had evolved to have enzymes that allowed them to deal with this toxicity. Let's consider those enzymes next. So here are some of the enzymes that organisms that utilize oxygen or can at least tolerate it, as in they don't utilize it but they grow in the presence of it. These are the enzymes that they use. They use catalase, which is able to convert hydrogen peroxide to water and oxygen. They use peroxidase, which uses the reducing power of NADH to convert hydrogen peroxide to water and NAD+. Okay? They use superoxide dismutase, which takes these oxygen species, a proton or protons, and converts them to H2O2 plus oxygen. Typically, this is only helpful if it's used in conjunction with catalase. That's what this option down here is showing. So that system is utilizing some additional protein components that we won't discuss here, and some have something called superoxide reductase. So the point here is that we have a suite of enzymes that neutralize toxic oxygen species. If you don't have one or more of these enzymes, then you cannot grow using oxygen and you cannot grow in the presence of oxygen. And so the presence or absence of these enzymes is really key in determining how organisms grow, what their pattern of growth will be in response to oxygen levels in the environment. All right, so how do we describe microbes from the perspective of their oxygen use or their oxygen tolerance? This table breaks out the common ways that microbiologists use certain terminology to describe these relationships. Aerobes are able to grow using oxygen, and anaerobes are able to grow not using oxygen. But there are subcategories within those. An obligate aerobe can only grow if oxygen is present, and it will typically use aerobic respiration. Actually, it will always use aerobic respiration. A facultative aerobe does not require oxygen, but it can grow better with oxygen. The term facultative aerobe is often used synonymously with the term facultative anaerobe. As I mentioned previously, E. coli is able to use a variety of energy yielding processes. It can do aerobic respiration, it can do anaerobic respiration using nitrate, and it's also capable of fermentation, the third equation that I showed you on a previous slide. So it's a facultative aerobe. It could also be called a facultative anaerobe. Okay? Microaerophilic organisms require oxygen, but they like them at levels lower than atmospheric. So they like them at low concentrations of oxygen. They use aerobic respiration, but you could think of them using it more moderately than the true aerobic respirers, the obligates. In the anaerobic world, we have two categories. We have aerotolerant anaerobes and obligate anaerobes. Obligate anaerobes can only grow in the absence of oxygen, so oxygen is actually lethal to them. Okay? They'll use fermentation or anaerobic respiration as their lifestyle. Remember, anaerobic respiration is doing respiration, but using something other than oxygen as your terminal electron acceptor. Aerotolerant anaerobes do not require or use oxygen, but they can tolerate it. Most of those organisms that fall in that category use fermentation as a means of making their ATP. So those are the descriptions for various microbes in the context of their relationship to oxygen. Now, in practical terms, in the lab, we're often trying to characterize or understand the growth pattern of an organism in response to oxygen, and this is an experiment that you may or may not do on the microbes that you're isolating. You would set the microbes up in tubes of media, and the tubes would not be shaken, okay? And they would be incubated with caps. The caps are green and they're shown there, and those caps do allow oxygen to diffuse into the tube, okay? And what you see is this pink oxic zone, okay? It's pink because they're using a redox indicator called rizazerin, okay? And it's pink when there's oxygen present, okay? So we've got oxygen present in the pink zone. Just beneath it, we have a little bit of oxygen, and lower down, we're in the anoxic or oxygen-free zone. The A pattern, this would be an obligate aerobe, okay? The B pattern would be an obligate anaerobe. It's getting as far away from oxygen as it can. It can't grow anywhere where there's oxygen. The C pattern, this would be a facultative aerobe. Some books will call that a facultative anaerobe. And I know that because it's growing better where there's oxygen. There's more cell mass there, but there is also cell mass through here. Okay, so it can grow with or without, but it prefers to grow, or it grows better, in the presence of oxygen. The D tube is a micro aerophile. So it's growing in the presence of oxygen, but preferentially where you have low concentrations of oxygen. In the E tube is what's known as an aerotolerant anaerobe. It's growing throughout the tube, basically ignoring the different concentrations of oxygen present. It's growing the same way throughout the tube. So with that, this concludes our discussion of the environmental factors that affect microbial growth.